my name's Mary. I am the Ecology Education Manager here at the Civic Garden Center. We're located in Cincinnati, Ohio, and so if you're local, please come and visit us. We're in the Avondale neighborhood. And if you ever find yourself in Cincinnati, we're a really kind of tucked in secret that we'd love to share with people, so feel free to swing on by and pay us a visit. We've been here since 1942, teaching people about plants, and I designed this Botany Bistro series as a way for me to freely share the botanical knowledge that has meant so much to me over the years and that I've gained just from conversations, interactions, workshops, lessons that I've been through throughout the course of my career. And so today's Bite-Sized Botany is all about plant breeding and genetics. Last session we had to postpone because I wasn't feeling well, but I did record that session and it is posted on YouTube. So I talk about vegetative propagation. And so I won't be talking about aspects of vegetative propagation today, since we're going to wrap up our conversation and talk a little bit about current science and ideas for the future, because people are always planning something new. And so please plan to join us again in the new year. We're going to stick with our every other Friday schedule. And our next session is going to be January 12th at noon. We're going to be talking about paleobotany, which is pretty exciting. That's going to be a new topic for me. And so I'm excited to do that research and share what I find with all of you. We are always having a snack for lunch. And so my snack for today, or actually it's a nice healthy sized lunch, I've got some rice in here, I've got some beans, and this is sweet potatoes. I'm keeping with my seasonality right now, but the color of it is very close to the color of a plant that I'll talk about in our lesson today. So I just wanted to give you a little sneak peek of what I'm having. I hope that you're enjoying a nice healthy snack, healthy lunch on this day, and it's actually not too bad outside, so if you want to take this on the go, you can put your AirPods in and go for a little walk. I'll make sure that we get all the information to you. It's all auditory, so you don't have to worry about keeping the screen up. And then the other thing I wanted to share with you is your homework. We always have a little bit of homework. I try to do homework on the off weeks just so I can kind of share with you what is going on in my brain as far as all these topics go. And so tell us your stance on the future of plant breeding. So I think that this is one that some people feel very strongly one way or another about how we should be moving forward with the knowledge that we have gained scientifically over the last few years, really. It's a conversation that I hear echoes of when people talk about AI and how that's changing the way that we do some things in our society. And so we'd like to hear from you. And, you know, I'm always open to conversation. It's important for us to have good discourse and learn from people that don't think exactly the way we think. Because if you get caught in that bubble, sometimes you can eliminate good conversation. So please feel free to share your ideas with us on social. And I'll be putting up a post as well next week. All right, well, let's dig into it. Hope everybody's ready. I'm ready. This is going to be just a little bit of a review at the beginning on what genetics are and kind of what we discussed in session 1 of this particular part of the series. And then I'm going to be talking about particularly disease resistance in agricultural crops and how that breeding and those programs have resulted in some amazing varieties that we can use, some good stories that we can share. I'm also then going to talk about how else we have been able to alter plants, including nutritional value and salt tolerance and a couple of other things that we'll discuss there too. But all of this happens very slowly. So if any of you are interested in this work and you are eager to learn more or become part of the important work of plant breeding and plant propagation programs, there's space for you. There's time for you. So make sure that you find a good program around you that you can work and get started on that. But there's always space for that. Some people work their whole professional lives on a plant product that won't be released for decades after they retire. And so this work does take a lot of people working together very hard. All right, so let's start off with a little bit of a review of genetics. So I'm going to be throwing out some terms. DNA, it's inner cells, it's also in plant cells. And that really is responsible for having the information for coding what is going to be your cellular structure and the way that your body is built. Genes are segments of your DNA that give you unique physical characteristics. And then also we have these different inputs from both of our parents. So because we were created sexually in sexual propagation, then we have half sets from both of our parents, so that we're able to be a unique individual with our own combinations. Whereas on the other hand, asexual or vegetative propagation, which I discussed last time, is a genetic identical copy. So we don't have the mixture of those different chromosomes and different parts of our DNA sequence. And so it should be an exact genetic replica if you're doing cloning. And so when we think about plant propagation, traditionally it was using, different kinds of pollen. We were making hybrids. We were putting these things together, and then we were saving that seed. We were growing that seed, and then we were growing additional generations of that plant to kind of find out, okay, does it still have the characteristics of the hybrid population, or does it revert back to one of the parents over time? Obviously, this takes a long, long time, and lots of good equipment, lots of controls, but these breeding programs can be, like I said, generational. And so there's nothing wrong with that. That's what we've been doing for a long time. But in the fairly recent future, we've started to accelerate that process by different methods, which we'll discuss a little bit today. But let's back up a little bit. And we're talking about what we actually are taking advantage with our scientific advancements. So we're taking advantage of genetic mutations or differences in how the cells are replicating that DNA when we divide or replicate our cells. And so sometimes it can be an exact copy of your cell, but other times there can be little mistakes. And so you could have a deletion, or you could have a replacement. And so these little errors are called genetic mutations. And a lot of times these mutations don't affect our bodies significantly, but sometimes it can be very challenging to overcome these mutations, and that is why our plant family struggle with these particular diseases or pathogens that we'll discuss. They do maybe need a little bit of assistance, a little bit of a leg up. So when we think about taking advantage of the genetic diversity that we have across this planet, and we honestly are just at the very tip of the iceberg with what we've actually discovered. I think there's out there, we find more things all the time. But we're thinking about the plants that are not going to be showing signs of an illness like other plants would. For example, we have most pathogens attacking a specific host. And so we can see this in the animal world. And so a lot of times we'll have, like bird flus or other things that they kind of stick specifically to a host. Occasionally we do read that these different pathogens and these different things can transfer between different species, and that is cause for alarm, right? And we're like, oh my goodness, these things are coming beyond their host, becoming non-host specific. And so in the plant world, we do have lots of pathogens that attack specific hosts, but we're able to take advantage of the resistance of non-hosts to be able to move forward with those plants. And so thinking about a tomato, for example, that is not going to be infected by the fungal pathogen that causes rust on corn, but can be affected by, other kinds of wilt that corn might not be affected by. And so this resistance we can take advantage of. If we can isolate what happens inside the tomato that makes it resistant to that corn issue, then we can take that from the tomato and we can add it to the corn. And there's a possibility that that corn cell could uptake that particular marker and become more resistant to the pathogen that is attacking it. So that's the concept, right? It seems almost too simple when I say it that way. That, oh yes, you can just switch around DNA. There are lots of things to consider. There are lots of different techniques people have used. We'll talk about a couple of them today. And then there's also ethical concerns. So we'll mention those a little bit, but like I said, that's part of your homework, and so I think that's the conversation that we can discuss later. And so resistance is loosely defined as the ability of a plant variety to restrict the growth or development of a specific pest or damage it causes compared to susceptible plant varieties under similar environmental conditions. So we're really looking for plants that are strong, that are able to withstand stress and kind of, be sturdy and not succumb to these diseases that can often wipe out entire crops. Different fungal diseases, a lot of them have come up in our plant pathogen series, so be sure to go back and visit those. But yes, we have overcome some of these things before. I just want to reiterate, though, that sometimes the reason why we are caught in these situations where, oh my gosh, 90% of the crop has been lost to this pest is because we're growing the same exact genetic copy of something for acres and acres and acres, and there's not any resistance built up. And so once these monocultures become infected, it can be very devastating very quickly. And so we'll talk a little bit about biodiversity at the end of our chat today, but I just wanted to throw that in there. And so when we're looking at plants that have different resistance to these diseases, I'm specifically thinking about when I buy my plants in the spring. So hopefully you've got a couple seed catalogs that you're pouring through this time of year. I always like doing that on cloudy days, just kind of dreaming up new things I'm going to get for my garden. And so some of them you're going to see these different terms. One of them is high resistance. Another one is intermediate resistance. So high resistance describes a plant that highly restricts the growth or development of these pathogens. They can still show some symptoms and damage from the disease, but typically not as much as intermediate resistant variety or a susceptible variety, which would be kind of like your baseline with no resistance included. And so those are things that you might see. They might be abbreviated HR or IR. And then you'll see other initials next to the plant name on the plant tag. Some of them will relate to different types of pathogens. So for example, on a tomato variety, if you see VF after it, it means that it has resistance to two kinds of wilt, Verticillium and Fusarium. And so those two types of wilt it will be resistant to. It might still show some symptoms, but it is much less likely to succumb to that wilt over time. And so it doesn't mean that it's going to be resistant to all diseases, though, right? It has those specific markers in it that say that, oh, yes, it can withstand this particular wilt, but maybe not a leaf disease, so we just have to pay attention to what we are shopping for. And I want to make sure that you know that those little initials can mean things. And so be sure to look those up, especially in my garden. I find that I really need to get those resistant varieties. And so I don't want to buy a plant that isn't followed on the tag with a couple of initials there. It's always something that I'm looking for. And so how do they put these together? So I did stumble across a pretty good explanation on a website. I know that some of this can be a lot of science, which I appreciate, but also get lost in. And so I think they kind of put this together really well. So I'm going to share from this website what I found. So a lot of these resistance types create something that agriculturalists need in order to be able to be productive and know that they are going to produce a full crop. So when they are asking for something that is resistant, how do they actually do that? And so they will find plants, could be in nature, that have specific mutations that show resistance. And so what they'll do is okay, great, like this one didn't succumb to this pathogen in this greenhouse environment. And then they'll try to do that trial out in the field, and they'll see if it also translates out into the field. And then after that, they'll start breeding and crossbreeding and crossbreeding and crossbreeding, that traditional method that I kind of talked about at the beginning with the moving of the pollen from one plant to another very intentionally and kind of studying the offspring of that and those different hybrid generations. But the problem that we have a lot of times is that we lose the quality of the initial plant that we were planting on purpose because of the amount of fruit it produced or the time from seed to harvest or the height of the plant. We have all this industrialization and mechanization of farming, and if you don't have uniform plants, it's very challenging to use those large scale types of equipment. And so I'm not saying, whichever way is better or worse. That's just the fact of how we feed our planet. And so thinking about how you are, incorporating that disease resistance, that's kind of the traditional way. But in this way, so this is more like gene modification. And so it's called R gene resistance. And it's also known as monogenetic qualitative or vertical resistance. It's usually controlled by one or a few resistance genes and can be more effective against some strains of the pathogen and less effective against others. The resistance genes, or R genes, work by allowing the plant to detect specific substances called effectors, which are produced by the pathogen. When the effectors are detected, the plant cell's metabolic defense mechanisms are activated, helping the plant prevent or suppress infection by the pathogen. And so this typically happens it's a little bit of an oversimplification. This is like a gene for gene model. So if it has the correct R gene, then the pathogen is creating this effector, then it's able to kind of identify it, just like our own body with our white blood cells, getting ready. After we've had a vaccination, it's like, oh, I can recognize this. I'm going to start to either have cell die off in that area, kind of self-quarantine on the plant. I'm going to start producing different types of cells, different types of proteins that can, kind of block this or create a callus. So it's just that kind of signaling that will happen in the plant's head if it's able to identify what's happening with the pathogen and as the pathogen is kind of going forward with the attack on the plant. And so I find this to be something that I appreciate, that plants are, very adaptable and very, flexible kind of. They're able to do things that we can't do as animal beings. They can regrow limbs and, overcome these challenges. But on the flip side of it, it's also made it easier for these plants to accept foreign DNA. And so I'm on this bridge of excitement about the possibilities, but also a cautiousness, I guess, I would say, of just how far do we go with it, and how do we proceed. And so something that people are doing now is to try to speed up the process of discovery. And they are using gene editing to do this. And so if you're constantly looking for new plants that have new resistance, you might not spend a lifetime exploring the world and all of the different ecosystems that have available plants. You might see, oh, this plant isn't succumbing to this disease, and it's right here. I'm just going to isolate some of its DNA, and then I'm going to introduce that DNA to the plant that doesn't have that, and, maybe it'll all be great. But I think that there's a lot to be learned still and a lot to be explored there to make sure that that's happening safely. But I definitely am not naive enough to think that people are not going to just do it if they can, because that's what people do. The other way that they're speeding up kind of the understanding of how these genes are being adopted into these plant cells is through marker-assisted selection. And so you can find a little mark on this genetic piece of information, and you're like, okay, when I see this sequence, I know that that protein is going to be coded for and that it has the R gene that it needs now. And so you can do that from a small amount of tissue culture. You could analyze parts of the seed germplasm for that. You wouldn't need to grow generation after generation after generation. So things that took, maybe 50 years are taking more like 10, 15. But then a lot of things have not gotten out of the deregulation phase. And so we still have several products that have not come to market based on just the need for them to be kind of studied more and then generally accepted by some of our regulatory agencies. And so let's think about a real life example. So I mentioned that I have some sweet potatoes. They smell so good. They're in season right now. And so this looks a lot like a plant that is much sweeter and tropical. And I thought about getting some just for today, but it isn't in season. And so I wanted to make sure that I was being a good role model. But that's papaya. And so I do enjoy some dried papaya from time to time. Most of the papaya that we eat is grown in Hawaii. And so there was a challenge for papaya. And, you know, it's not totally gone, but there are methods to handle it now. And so we haven't talked about papaya on this series yet. And so I'm excited to share with you this story. And so in 1945, the first report of this ring spot virus was given. And so it was first discovered in Oahu and caused such severe damage to the papaya industry that the whole industry was relocated to another place just to try to get away from this pathogen. And this was in the 50s and 60s. It was all relocated. They expanded in Pune because this virus was absent. However, in 1970s, the virus was found about 19 miles away in Hilo, and the Hawaii Department of Agriculture took to removing infected trees and quarantine to keep it from spreading. And so we know how quarantine works. It works for maybe a little while or it slows things down, but it doesn't stop things altogether. And so there were some efforts in 1986 to start to develop a virus-resistant transgenic papaya that could transform the current lines of papaya with a protein that has a resistance for this virus. And so, and about 6 years later, they were able to identify this line that showed resistance under greenhouse conditions. And so then it was a few years later, in 1992, the papaya ring spot virus was discovered in Pune, where 95% of Hawaii's papaya was being grown. And so it was really important that we had some options, or else that whole entire industry could have gone to the wayside very quickly. The experiments for the resistant variety were being done on Oahu because that was where the virus was already. And so it was just right on time that as the virus spread to the planted fields, they were able to be also testing this other resistant variety. And as they were replacing things, they were able to stay caught up. And so even though it did lower the exports by about 50% for a couple of years while they were getting everything organized, the resistant papaya is the one that we typically will have a lot of times in the store. It's actually called, the variety is Rainbow, which I think is fun. And so honestly, without the development of this particular disease-resistant variety, papayas would be very, very expensive and much more of a delicacy. And so if you enjoy papayas, some of our other tropical fruits, make sure that you're eating those as ethically as you can, in season if possible, but really trying to promote the farmers that are doing this hard work of trying to preserve the genetic diversity that we have, increase the genetic diversity that's out there, and protect those species that we all have grown to love. So that's an interesting experience in Hawaii. I'm going to talk now about one that we have mentioned before, and I'll be going more into biotechnology in April. We have a couple of sessions there that are biotechnology focused, so I'm not going to talk too much about biotech right now. But golden rice is something that a lot of people might have heard of, and so that's another genetic modification. And so whereas the papaya was kind of like, oh my gosh, we just need to save the crop right now, Golden rice is being developed as a way to add beta carotene into a staple crop that many, many, many millions of people eat around the world on a daily basis. And just trying to improve the nutritional quality of that food so that the people that are eating it, sometimes exclusively as part of their diet, are getting all of the nutrition that they need. And so the reason why it's called golden rice is that the introduction of this beta carotene actually gives the grain a golden color. We love to add turmeric to our rice at home, so it kind of looks like that when you see it. And so it's, the identical beta carotene that's found in green leafy and yellow colored vegetables, orange colored fruit, and gives it that same color. Now, there is still some some ethical issues that people are working through on, is this okay? Is it going to cross with other crops? Is it going to reduce the genetic diversity? What is going to be going on with it? And so this has not been as widely planted yet as hoped, but possibly we'll kind of keep our eye on golden rice. Cassava is also being worked on to be added with beta carotene, iron, and protein. And so both of these projects are using a range of breeding techniques, transgenic approaches, genetic modification. And so I feel like after talking about a few of these, you're probably ready to go, right? You're like, let's do it. How do I get on board? Well, you can go out and get yourself a gene gun, and it's kind of exactly as it sounds. GENEGUN, a gene gun. And this was developed in the late 80s, and it delivers external DNA to cells. And so basically you have an elemental particle of heavy metal coated with DNA, and it's able to transform nearly any kind of cell. It's not limited transformation of the nucleus. It can also transform organelles and plastids. And so the original gene gun was an air pistol, which was modified to fire tungsten particles. So I just find this to be very curious that someone's like, well, let's shoot it with DNA. I don't know. And they created a gene gun that was actually fairly effective. And so they used it first on onions because they have a large cell size, and they were able to deliver particles coated with a marker gene. And so it's also known as biolistics, and it's proven to be a very versatile method of gene modification. You can buy your own gene gun for anywhere between $10,000 and $30,000. I did see one used on eBay for like $2,600, so I don't know if that's working or not. But these types of technologies are becoming more and more widely available. And so it's just something to think about. This isn't something that only happens in labs at very fancy universities. It's something that could be happening in your neighbor's house if they're going to invest in something like that. And another method is CRISPR. So some of us have probably heard of CRISPR, just in passing, a random conversation. But this is a method that's been used for just over 10 years. And it stands for Clustered Regularly Interspaced Short Palindromic Repeats. That just rolls right off the tongue. But they're repetitive DNA sequences, and were reserved in bacteria. So there's an interesting parallel here where we were like, oh, something in nature is doing this well, and we want to copy it. Biomimicry, right? And so we were observing that these spacer DNA sequences repeat the viral sequences. And so it was discovered that the bacteria that transcribe these DNA elements to RNA when they are infected by this virus. And so you can have the RNA guide a nuclease, which is a protein that cleaves to DNA, to the viral DNA to cut it, and then it stops the replication of that virus. It protects against the virus. And so these nucleases are named Cas for CRISPR associated, or CAS. And so then using Arabidopsis, which is like the superstar of the plant experimentation world, was used to be able to think about how can we be cutting the different types of viral DNA, how can we add resistance into these different plants. And so it's just been really neat to see how they're able to transform rice, tomatoes, and it's very effective. They are able to identify different parts of plants with a lot of information. So it's not like single changing of these genome sequences. You can actually change multiple parts of them, replace multiple genes, or have them expressed in different ways. And you can do that with this CRISPR method. And so the guide RNAs can be made so that it's specific to only one sequence, and it improves the chances that the DNA will be cut at that site and no other part of the genome. So you're not taking a risk on losing some of the characteristics that you really like in the plant as much. I mean, that can still happen, but you're hopefully able to preserve some of those things that you really like. And so we talked a little bit about just protecting our crops from disease. We talked about possible nutrition additions. We talked about a couple of methods. Something that people are working on and hasn't really been able to be translated outside of the lab yet is salt tolerance. And so as we think about the stresses that we might be dealing with in the future, we have sea level rise, we have saltwater intrusion to some of our freshwater areas. And so being able to grow plants in salty water or salty environments is going to be something that's going to be fairly important to many people around the world moving forward. And so there have been some greenhouse experiments that have been working okay, and it's just a little bit harder to replicate those out in the world when there's a lot more variables. But some of these are actually promoting a protein that's called an antiporter. And so this antiporter accelerates the exchange of sodium and hydrogen ions across a cell membrane of a plant. And when the sodium is absorbed by the plant, it will disrupt the enzymes in the transport of water. And so you need to be able to get these antiporters into the plants more so that they can take out that salt. And so with these additions of these antiporters in other plants, scientists were able to breed plants that could grow in water a third as salty as seawater with few ill effects. So it's not straight seawater, it is diluted, but they didn't have as many die-offs as they would if they didn't have these antiporters. They pushed the sodium ions into vacuoles, which are sealed off places within cells. And these actually occur in natural halophytes, which are salt-loving plants. Vacuoles can become so big, they're called salt bladders. Quinoa is an example of this. It has tiny little bladders that look like little spheres. And so they are removing that salt from their system. So I'm really excited to see where that research can go. The humidity in the greenhouse is also a good helper. So it's about 40% humidity in the greenhouse, where it's only like 5% outside in the field. And so really thinking about how to control some of those environmental factors has been a challenge. Other things I am interested to see coming down the line are edible vaccines. Right now, the challenge with these edible vaccines is that they need to be typically in cold storage or very carefully transported. And so until they are being able to be grown exactly where they're needed, that might not be something that's going to be coming around for your flu shot next season. But the concept of being able to eat, a few leaves of spinach and getting, a child vaccinated against some of these childhood diseases or other things that could be very detrimental to their development. I mean, I find that to be, pretty wild. It's not science fiction. It's happening. Or at least the concept is there and people are working on that. And so what I really wanted to end with is just, on the positive note of human innovation and, just like this constant seeking for understanding of our world, that is impressive, and I think does have inspiration for me to continue to explore and go farther. However, at the end of the day, what are we talking about here? We're talking about biodiversity. We're talking about a need to be able to identify lots of different ways for handling stress, lots of different adaptations that plants can share with us that we need to take advantage of if we're going to continue to live on this planet. And so having respect for biodiversity is just inherent. It's just part of all of this. And so you don't want to harm biodiversity by putting something into the environment that will have detrimental effects. You don't want to take advantage of the benefits of previous biodiversity by just scraping all the topsoil off and planting a monoculture and then wondering why I have to put on so many synthetic fertilizers or pesticides. We can't ignore that. And so what plant breeding programs have done, and a lot of times what I feel like we're moving forward, is we're trying to create biodiversity on a very quick timeline. And it's not to say we can't do it safely. I don't want to stand here and say that I know exactly how this is all working, because I don't. But at the same time, I feel like, how fast should we go with it? But what should we really be looking at that our world needs moving forward? Nutrition, medications, drought and disease tolerance so that we can continue to feed people. All of these things are going to be so important moving forward. So I think that I'm just putting a little call out to everyone there and saying thank you for being part of this journey with me. And I'm really inspired by what I read when people are trying to think differently and saying, okay, I understand that this part of our country is not going to be adaptable to being able to grow some of the food we've usually grown. So how can we get ahead of that? But we also don't want to treat a symptom, right? We want to make sure that we're also looking at the cause. And so eating seasonally, eating less meat, making sure that you are trying to decrease your personal footprint, and then encouraging others to do the same, your workplace, where you worship, where you go to school, and just always be thinking about that. These are amazing advancements that we have seen. And really, who knows where we're going to go from here. But at the same time, let's make sure that we are preserving the biodiversity we already have and valuing that for really what it is. So thank you all so much for coming today. I just wanted to remind you that our next session is going to be about paleobotany in the new year. I hope all of you have a wonderful transition to 2024. And I'm going to open up the chat if you have any questions. Really happy that you joined me for our last session of 2023.